Hi y'all. This is going to be Nervous System 1 lecture, and it's kind of a long lecture. And so just stick with me, and I promise you that as we get through it, everything will be easier. And so, as I said, it's Nervous System 1, so we probably have covered some of the nervous system already. I know we covered it with the muscular system. We know that with the nervous system and histology that we have neuron is the main communication cell. It has dendrites, it has cell bodies, and then it has an axon. And we know the dendrites, there's usually many of them, and it's for increasing surface area, whereas the cell body is responsible for making decisions, and it has like most of the organelles that are there. And then we know the axon, there's usually one of them, and then that's where the responses get sent out, and then the axons branch, into these axon terminals, right? And that's where neurotransmitters release, like acetylcholine with the muscular system. So we're going to go ahead and move forward. And we know that the main function of the nervous system is going to be communication. It works with the main other controlling system of the body, which is the endocrine system, which is by far my favorite. And we know that it controls homeostasis, which means when things are supposed to be in this zone and they get out of it, the nervous system and endocrine system bring them back together. And we know that one of the main differences between the nervous system and the endocrine system is the nervous system works super fast. Its responses are usually always super fast. And we know that it works by means of an electrical signal, and all that means is we have sodium potassium pumps that are going on and off through the dendrites, cell bodies, and the axons. And so the three functions of the nervous system follow the same three functions of the neuron that we see here in this picture, is that we know that they pick up sensory input, and that's going to be the function of the dendrites. And we know that it picks up information from outside the body and inside the body. And then it takes it to the processing center, which is going to be the cell body, and that's where decisions are going to be made. And then if a response is needed, like if I put my hand on a stove, then the response is going to be taken out through the axon. And that's what we can see here in this picture. And then it usually is going to go to a gland, or it's going to go to a muscle. And so we kind of think about this in terms of what we do on a daily basis, right? We have sensory input coming in that can come in through a neuron that's blue, and it goes to our spinal cord or our brain that's green, and it decides, oh, we see a glass of water. Are we thirsty? Yeah, we're thirsty. We should probably drink. So that neuron sends a signal out through ways of the motor pathway, which is going to be motor for M is the same as movement, right? And that's going to go to our bicep, and it's going to cause our bicep to pick up the glass, right? And so it's kind of the same function. So the really weird thing about the nervous system is that the cell does the same things: sensory input, decision, which is integration, and then responses, right? But then we have neurons that do these same things as well. So the nervous system can always be kind of divided into these three main functions, and the three main functions can occur in one cell, or it can occur through many cells, which and the cells here are neurons. And so the organization of the nervous system is that we have the central nervous system and we have the peripheral nervous system. The central nervous system is going to be the brain and the spinal cord, pretty much anything that's going to be protected by bone. And then the peripheral nervous system is going to be anything that is outside that's not protected by bone. And that we know that the goal of the peripheral nervous system is to basically gain information and communicate and cause responses that the central nervous system controls, right? And so when we say white matter and gray matter, it just means that the tissue of the nervous system is going to be two different colors. It's going to be rather white, or it's going to be gray. And the reason that something is white is because that the axons that we talked about in histology, the axons have myelin around them, and myelin is that protective sock that covers the axons, and myelin is made of fat and proteins, so lipids and proteins, and the lipids are going to be white in color, hence it's white matter. And then gray matter is going to be gray because there's not as many myelinated axons present. So what just some words are to kind of help you. A ganglion, which we might have talked about already in terms of the spinal cord. A ganglion is going to be an area where a whole bunch of cell bodies collect, right? So if you have a whole bunch of sensory neurons coming in, you're going to have a whole bunch of cell bodies that are there, and they're all going to kind of just be in a general area, and that's what a ganglion is. And the peripheral nervous system is a lot more complicated. It has sensory input coming in, but then it also has motor responses coming out, right? And so the way that we kind of remember it is that a sensory neuron is also called afferent, and a motor neuron is also called efferent. And I always remember it, A comes before E, so that's somewhat helpful. And then the efferent always goes to something called the effector, which is a muscle or a gland. And we can see in the bottom right picture that we have the spinal cord. And the spinal cord is going to have a ganglion, which is that big bulbous swelling that we see in the kind of the bottom right. And that's where there's going to be a cluster of cell bodies that are going to be there. So the word somatic always means usually things that we're aware of, like our joints or skins and our muscles, whereas visceral are going to be thoracic and abdominal cavity, things that we really have no idea that's going on. And so when we think about the somatic nervous system is going to be something that controls mostly skeletal muscle and things that are going to be voluntary and under conscious control, right? And so when we mean that, it can be joints, skin, muscles. And the deal is interesting, though, is that hot stove reflex that we've talked about so many times, that we are aware that our hand is moving, but we're not actually kind of in that situation. We're not necessarily making a conscious decision to move it. Your kind of central nervous system takes over because it's really a dangerous thing that can happen to your body. Whereas the autonomic nervous system are things that are visceral, things that we're not aware of. So smooth muscle, cardiac muscle, our glands that secrete saliva in our mouth, right? Things that we're not aware of. And so, this picture that we see here on the right here is that we can see that we have a picture of our skin, our integumentary system on the right, and that is going to be sending signals from things that we're aware of, right? That goes to our spinal cord and causes muscles to contract. That's going to be the somatic part that we're aware of. The picture on the left is going to be something looks like our intestines, right? And that's going to be things that we're not aware of. So it's important for us to remember that the nervous system, the spinal cord, is processing things that we're aware of and also things that we're not aware of. The autonomic nervous system is the thing that is always kind of freaky. This is the responses that we're not aware of, and it's called the autonomic, and I always think about it being the automatic system, things that happen automatically. And so the sympathetic nervous system are the things that are going to cause us to be in our fight or flight moment, like I'm being chased by a bear. A long time ago, a professor in psychology was trying to teach me literally a little bit about like how the brain works and the nervous system, and she taught me that in psychology, the way that she was taught it was that the sympathetic nervous system is kind of your oh shit mechanism, right? It's the fact that, oh my gosh, this is really, really bad, and your body's kind of freaking out, and your body's got to do something to prepare it to be okay, right? And so when you're being aroused, when you're being chased by a bear, your reproduction, your urinary, and your digestive systems are going to slow down because you don't want to say, bear, hold on a second, I have to go potty, please don't eat me, or hold on a second, I need to eat, right? And so the deal is those systems slow down, whereas kind of every other system becomes heightened, whereas the parasympathetic is your calming effect. And in that situation that your body's in, digestion, urinary, reproduction are all going to be elevated, right? Because you have time and it's not going to be detrimental to your well-being if you eat or you go potty, right? And so the picture on the right kind of takes us through what happens when each system is going on. Is it heightened or is it going to be slowed down? And so that's the automatic part, the things that we're not aware of. So when we're being chased by a bear, we're not aware of that our pupil opens up, right? So more light gets into our eyes, so we're not limited by light. So I kind of found this picture online, and it was really helpful from a flow chart standpoint, that we have sensory input coming in, right? That sensory input can come from like anywhere, and that's in the peripheral nervous system. The peripheral nervous system then takes things to the central nervous system, and then the central nervous system decides if a response is needed. If a response is needed, it goes back out through the peripheral nervous system, and it goes out through your motor, your movement pathway. Your movement pathway can break down into one of two things. It can do things that somatically that you're aware of, or it can be the automatic thing that you're not aware of. And the things that you're not aware of can cause your body to kind of be in your heightened sympathetic mode, or it can cause you to be in your calming mode. And so if people thought that that diagram was more busy, I came up with kind of an outline form to kind of help you, and y'all can see that the peripheral nervous system clearly looks like it's doing more things, right? It's a lot more complicated in the terms of it's dealing with sensory input and motor responses coming in and out. So now if we go into the histology and the cellular things, this is where things get to be a little bit more complicated. But what basically it is that the nervous system, we've talked about neurons, right? Those are the cells with the dendrite cell bodies, and axons, the parts that are going to be doing a lot of communication. However, there are these supporting cells, and these supporting cells, which we did talk about in histology, are the ones that are really, really important because they're the ones that help the neurons do the job. So we talked about this in terms of President Vaughan at the university. He's going to be the neuron, but then he has then all of his supporting staff around it that help him do his job. And so the ratio can be 9-to-1, 50-to-1, so it can be variable depending on where the neuron is, but each neuron has a whole bunch of supporting cells, and these supporting cells are going to be called neuroglia, and there are going to be 4 different types that we're going to talk about in the central nervous system, 2 types in the peripheral nervous system. And the reason that they're called neuroglia is because neuroglia means like nerve glue, and it's the fact that they stick to the neuron and they really help the neuron do its job, right? And so without these neurons, without these neuroglia, the neurons simply just can't do their job. So I kind of tried to break this down into the situation so that there are types of neuroglia. There's 4 types in the central nervous system, 2 types in the peripheral nervous system. And so we can see that the 4 types in the central nervous system are going to be oligodendrocytes, epidermal cells, microglia, and then astrocytes. And then the 2 types in the peripheral nervous system are going to be the Schwann cells and the satellite cells. So the 4 types of satellite cells, the supporting cells in the nervous system, specifically the nervous system, we're going to talk about those first. And so these are the 4 types of neuroglia in the central nervous system, right? And remember that we didn't identify these in histology, and the reason that we didn't identify them in histology is that they're even smaller than the neurons, so they're really, really, really just hard to see. And as we can see, that because the ratio of neuroglia is so much higher than neurons, they make up a lot of the brain mass. So the first type is going to be astrocytes, and I know this slide is like overwhelming, right? But it's because astrocytes are the most abundant. They make up most of the tissue in the brain. They're called astrocyte because they're star-shaped. And what they do is they unite the neuron with blood supply, which means if my hand is the neuron, the astrocyte holds on to me, but then it also holds on to a capillary bed right by the neuron. And so what does it do? It keeps the blood vessel near the neuron, so the neuron has access to the nutrients it needs, right? So that makes total sense. The second thing that the astrocyte does is that it makes sure that the environment all around the neuron is okay. So it makes sure there's not too much sodium or potassium or calcium, which would totally mess up the sodium-potassium pumps, right? It makes sure that any neurotransmitter that's around it, that it can rather reabsorb it or kind of get rid of it. I'm sorry, I'm having something in my eye right now. And it also can control what is in the blood, right? So it's really, really important because it helps keep the environment perfectly clean all around the neuron. The other thing that an astrocyte does is that astrocytes communicate with each other by gap junctions. And so what they do is that astrocytes are connected to each other by gap junctions. So if this astrocyte has too much sodium potassium around it and it needs to do its job, the other astrocyte that's right by is also going to know that, hey, you're probably going to need to clean up some sodium or potassium over here as well. So the other thing the tight junction astrocytes do is that they form tight junctions. Remember, tight junctions are like a zipper. We talked about these with the integumentary system, and tight junctions act as a zipper. And what does the zipper do in your hoodie is that it makes sure that like the cold air doesn't get inside of you. So what does the zipper do is it makes sure that things don't get places, right? So it forms tight junctions. And it forms tight junctions around these capillary beds, because what happens is that because your blood changes so much and it's always changing, the brain can't really handle all those changes. So basically most of your brain does not have contact with blood, and we'll talk about this later. So what it does is it makes sure that there's a zipper all around those blood vessels so no blood gets into the brain and causes it to be like not happy because the blood is changing so much. And so it forms those tight junctions. The other thing that astrocytes do is that they can somewhat help neurons be repaired if they're damaged. And most of the time, neurons can't be necessarily repaired, but they can maybe prevent more injury. They can stabilize an area, right? So astrocytes are super busy. So just to kind of recap what they do, the first thing that they do is they unite neurons with blood vessels, right? The second thing that they do is they keep the environment all the way around the neuron healthy. The third thing that they do is that they talk to each other. So they talk to each other through gap junctions. The fourth thing they do is they form those tight junctions that prevent blood from coming in contact with the brain. And then the other thing that they do is they can also help neurons grow and somewhat be repaired. So when we think about what an astrocyte looks like, several of these pictures have it. So if we look at the top left picture, the pink picture, the arrows are pointing to astrocytes. They're very star-shaped. We look at the top right picture, we can see again astrocytes being very star-shaped. Bottom right, we can see the neuron, which is green, and the astrocytes, which are star-shaped, which are going to be yellow. If we see it like that. And then what we do is that we look at the left picture. In the left picture, you're going to see the neuron on the bottom left, and then you're going to see the capillary bed, which is the circle, and then you're going to see the astrocyte, which links them together. So the second neuroglia cell we're going to talk about is going to be the microglia. These guys are crazy important. And you wouldn't think that. They're the least numerous. There's the least of them. They're the smallest, but they're called microglia because they're so small. They're thorny. What they do is they act as a macrophage. And a macro means big, phage means eating, so it's kind of a type of a white blood cell. And basically what it does is it can eat any damaged tissue. It kind of acts as a janitor. It cleans up the brain. And so the reason that microglia is really cool is that it's one way that doctors can tell if someone has had a stroke is that the presence of microglia indicate a stroke. And microglia can also release proteins that are like a byproduct of them cleaning things up that can be picked up in the blood. So when we look at these pictures, especially the one in the square, we can see that the size of microglia is really, really small compared to the astrocyte that we've already talked about or the neuron. So whenever we get to, again, these pictures, we can see astrocyte and microglia. Remember that microglia help clean things up. They're super important. So then we get to the third type of neuroglia, and that's going to be your epidermal cells. Epidermal cells are going to be cuboidal in shape, so they're not looking like all these other long star-shaped cells. And so what they're going to help do is, because they're going to almost look like cuboidal cells with cilia, is that they're actually going to help make and move cerebral spinal fluid. So what is cerebral spinal fluid? When I told you that the brain, most of the brain cannot have contact with blood, you're thinking, well, how does it get its nutrients then? It's because certain cells help make this perfect fluid called cerebral spinal fluid. And this perfect fluid comes from the blood. And it says, oh, okay, you're calcium, yeah, you can come over. Okay, yeah, you're sodium and potassium, yeah, you can come over. Uh-oh, you're a nitrogenous waste, you can't have contact with the brain. And so the epidermal cells help make this perfect fluid that's called cerebral spinal fluid. And cerebral spinal fluid can have contact with the brain, and that's how all the cells, like the neurons and everything, they get their nutrients. And so because they have cilia, they help move cerebral spinal fluid. And as I said, they look like cuboidal cells. And so what's going to happen is inside of your brain, you have these rooms, and these rooms are called ventricles. And inside of these rooms, basically what happens, it's going to be lined with these epidermal cells. They're going to help make cerebral spinal fluid, which is going to be in the space, and then the cilia is going to help move the cerebral spinal fluid through all of these different rooms, right? So it's kind of like going through a maze. You have all these different rooms, and the cerebral spinal fluid gets moved through all these different rooms, right? So when we look at epidermal cells, if we look at the bottom right first, we can see that these big purple cells, they have cilia, they look like cuboidal cells. If we look at the top left, we can see that there's an opening, and that's where cerebral spinal fluid is. And then the epidermal cells are going to be the brown cells that line that cavity. And then we look at the top right picture, you're going to see in the center, it's going to be where it says central canal. That's going to be where cerebral spinal fluid is. The central canal is actually in the spinal cord. And then what we do is we have epidermal cells that line it, which look very much like simple cuboidal cells that we saw in histology. And so just to kind of make sure that everybody's good, this picture in the top right really has helped me a lot. That what happens is that we can see that we have a blood vessel. It's called a blood capillary. And then what happens is that we can see that there's an arrow that shows cerebral spinal fluid coming out of that. And then we see the epidermal cells, which are the cuboidal cells with cilia, are going to be there to help form cerebral spinal fluid, and then the cilia are there to help move it. And we can see that there's the word ventricle, which means space. So that's going to be that space where cerebral spinal fluid is, and then the epidermal cells are going to help make it and then move it. The fourth type of neuroglia in the central nervous system is going to be oligodendrocytes. So oligo means few. Dendrocytes are going to be very similar to Schwann cells that we talked about in histology that are going to form myelin. So what happens is that with oligodendrocytes, you're going to have the neuron right here. Then an oligodendrocyte is going to reach out with one of its processes, and it's going to wrap, for example, the axon and form the myelin sheath. So it's not individual cells that wrap, it's kind of the cell processes that are going to wrap. And what I mean by cell process, I'm talking about the axons or the dendrites. And so what it does is it creates a myelin sheath in the central nervous system. And we've already talked about how myelin is going to be white, right? And then we've talked about also how we've got nodes and internodes. And we talked about this actually in lab, right? So the nodes are going to be the gaps where there's not going to be any sodium potassium pumps and there's not going to be any myelin. And then that's how the signal, the axon, the action potential is going to travel down the axon. So I'll talk about this in a second when I show you a picture. So in this picture right here, the top right, you can see that you have an oligodendrocyte, and each of its cell processes wraps and forms the myelin sheath, maybe even around a different axon, right, as we can see here. And then what happens in this top left picture, we can see that where the myelin is, right, that there can't be any sodium potassium pumps, because where the myelin is, you can see that, oh, it doesn't even have any contact with the outside environment because this oligodendrocyte has wrapped it so tightly. So where are the sodium potassium pumps? It's going to be in these spaces right here, the yellow spaces where there's not any myelin, and that's what we're going to call a node. So then we go to the bottom right picture, and what we see here is that we're going to have the nodes. The nodes are going to be the empty spaces, and what happens is that the action potentials keep jumping all around it, and that's what allows the myelin to allow the signal to travel faster, because instead of it having to travel all the way down, it jumps, and so that allows the axon to respond faster, for example. So we can see here we have epidermal cells, and then we can also see that the processes of oligodendrocytes are all wrapping individually the axons, which are yellow. So just to kind of review, the four types of neuroglia in the central nervous system is that, first of all, we have the central nervous system, right? And what happens, we have the neuron, which is kind of the brown color cells, and then we have the astrocytes, which are yellow, and the astrocytes are green, and we can see that they unite the neuroglia with the blood cell. They also form tight junctions to say, hey, no blood, come out of here. Then also we have microglia cells, which are the small ones, and they're the ones that pick up and clean up all the material. And if your microglia cells don't work, you can end up having lots of nervous system disorders that we'll talk about, I think, in presentation 6. Then we have oligodendrocytes, and you can see how each of the cell branches wraps to form the myelin sheath around the neuron. And then we have the epidermal cells, which help create cerebral spinal fluid, and the cells look like simple cuboidal cells with cilia. So all that is going to be the four neuroglia in the central nervous system, right? So if we kind of just make sure that we're good to review, we see that 1 is going to be the neuron, 2 is going to be the oligodendrocytes, which are going to wrap and form the myelin that we can see. We can see 3 is going to be a capillary bed. And then we know that 4 is going to be the axon, which is going to be covered in myelin from the oligodendrocyte in this situation. 5 is going to be the astrocyte. 6 is going to be the epidermal cells that are going to help create cerebral spinal fluid. And 7 are going to be the little small cells that are microglia that are going to help clean everything up, right? So those are the neuroglia in the central nervous system, right? And so one thing to kind of remember is that when students or kids under the age of 4 get a brain tumor, it's usually because there's a malfunction in the neurons, right? Because your neurons are still growing and developing in a child. However, when an adult gets a brain tumor, it's not because the neurons have undergone mitosis, because neurons usually don't undergo mitosis as adults. So what's really crazy is that brain tumors in adults are usually a function of there's a problem with the neuroglia cells, because the neuroglia cells start undergoing mitosis rates at a wrong rate, and that's usually what causes tumors in adults. It's usually not problems with the neurons in adults. So if we go over the two cells, the two types of supporting cells, or the two types of neuroglia in the peripheral nervous system, the first one are going to be satellite cells. And satellite cells surround the cell body, and what they do is that they make sure that the cell body is okay. They regulate the environment, they make sure that everything is okay, that there's not too much sodium, not too much potassium. They're very similar to the astrocytes in the central nervous system. And what we can see here is that we can see the cell body, which is really big in the bottom left picture, and then the satellite cells are these two dots that are around, and they just help the cell body. And again, in the right picture, you can see the cell body, which is really big, and then all of these small cells that surround it are going to be the satellite cells that are actually going to help the cell do its job and make sure that the environment around it is okay. So the second type of supporting cell in this peripheral nervous system are Schwann cells, which we've already talked about, right? Schwann cells help form the myelin sheath. We've seen them in lab. We've seen them in the lab video. They're similar to oligodendrocytes in the central nervous system, that they form the myelin sheath. They're individual cells. We know that the outside of the cell is called the neurolema. We know that there's no sodium-potassium pumps here because it's protecting it. So the sodium-potassium pumps are not where there's Schwann cells. We know that what happens is that the Schwann cells protect the neuron. We know that it allows the signal to occur faster. The other function of Schwann cells is it can also help clean some debris, which would make it a little bit similar to microglia. And so it can help clean up the area. So that's kind of important as well. And so cleaning up is similar to microglia, but because it can help the neurons not be as damaged and maybe help repair the neuron, that will kind of make it similar in function to the astrocyte. So when we look at these pictures, this shows how a Schwann cell is very different, like looking than a oligodendrocyte, and how it wraps, and that the outer coating, like we've talked about in lab, is the neurolema. So if we think about the neurolema, another name for the neurolema would just be the plasma membrane of the Schwann cell. That's pretty much similar, and then the myelin is on the inside, and clearly there can be no sodium-potassium pumps there because it doesn't have any contact with the outside environment. So in this picture right here, we can see how the action potential would actually jump because there cannot be any sodium potassium pumps here. And again, there's one nucleus because it's just a cell, and the outside covering is going to be the neurolema. So when we look at this picture, we can see what a myelinated axon looks like under the microscope, which I think is really helpful. We can see the axon and how that thick layer of myelin really does protect the axon, and how the neurolema is its outer covering right here. So we can see that we have a myelinated axon, and that myelin is really, really thick. Or we can also see how sometimes there are many axons inside, and sometimes like these axons don't have myelin around it. So that's kind of a little bit different. So y'all have seen satellite cells before when we were talking about different types of neurons in histology. So this right here is going to be a unipolar neuron. And the goal of a unipolar neuron is basically to make sure that all the information goes to this central nervous system, which is usually the spinal cord, and the spinal cord makes the decisions. So the reason that we would have a unipolar neuron is that we want to make sure that someone that's really in charge can process the information, and we don't leave like not as qualified people to make decisions. So in this situation is that we have information coming in from our skin, from our outside environment, which can be super dangerous, right? And that information is not going to be processed by the cell body. We're not going to trust that cell body. We're going to send it to the central nervous system. We're going to say, hey, this is what's going on, and you are powerful enough that you need to make that decision. So the cell body the signal just gets sent right on by. So the cell body does not process it. And so people are like, well, then what does the cell body do? Well, it still makes the neurotransmitter, it still has all the organelles, it just doesn't process the information. And so what happens is that you can see the purple satellite cells that surround the cell body that make sure the cell body is okay, and the cell body can still do its job in terms of everything else other than processing. So just to kind of make sure that we're super good, right? That the neuron are what we call the structural units of the nervous system, just kind of like the osteons were the structural unit of, which means the most important thing kind of the skeletal system. We have the neurons, right? And that they can conduct the messages. So really some things that are crazy about neurons is that they're really long. Like if we live to be like 100, we have cells that are like that old, right? That's kind of crazy considering how fast that our cells like on our skin regenerate. They're not mitotic, which means they're not reproducing and dying like cells in our skin, right? And that also they have a high metabolic rate because they do have sodium potassium pumps, which means they have ATP, which means they're releasing heat, and which means that they are functioning at a really, really high level. They need oxygen and they need glucose, right? So if we look at this picture, right, this is going to be a neuron. And we know that we have dendrites at the top, we have the cell body, then we have the axon at the bottom, right? And it's the same thing, sensory, processing, motor response, right? And so sensory, processing, response. And so what happens right here is that the sodium potassium pump, the signal is going to start in this region, the axon hillock. And then what happens is it's going to come down the axon. And then what happens, because we have individual in this picture, this is going to be a neuron in the peripheral nervous system because we have individual Schwann cells. And what happens is that the outer covering of a Schwann cell we know is called the neurolema. We know that we have nodes. And then what happens is that the nodes, the places where there's not any Schwann cells, are the only places that we have sodium potassium pumps. And it's going to leap. The sodium potassium pumps are going to leap and it's going to go super fast because it doesn't have to go slowly all the way down. And then we have the axon terminals, right? So hopefully that's super clear. So remember that when we talked about sensory, processing, motor, it's the same thing when we look at neurons. We have sensory neurons, afferent going in, interneurons in the central nervous system processing, motor neurons, efferent neurons coming out, going to a muscular gland, causing a response. We kind of keep having the same recurring theme. Sensory, processing, response, right? So it's the same kind of process. So with that, I hope that that helps you with the nervous system, and I super hope that you're not totally confused with all of these different neuroglia cells. And if you have any questions, you know I'm always available. Thanks a bunch, guys.